Hi, name is Paint number 11, and today I'll talk about chapter 5 to chapter 8 from biology. So first we're going to learn about the membrane structures. So it is the phospholipids that arrange in the bilayer, and it is called as the fluid mosaic model, which it was called fluid because of the phospholipids. It was not because of the phospholipid is not a solid. It was kind of like solid liquid things, so that's why. And how did they visualize the membrane? They visualize it by they visualize it by freezing the cells so that they can cut slice of the membrane of the cells out because it's have been freeze like that. So the cellular membranes has in totally 4 components. The first one is the phospholipid bilayer, which is this one, the one that we have learned in the midterm, that one. So second one is the transmembrane proteins, which is the integral membrane proteins. Third is the interior proteins network, or is the per, 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 peripheral or the intracellular membrane protein, which is these ones that was coming outside from the phospholipids by layers. The last one is the number 4, the cell surface markers, which is this one. It is the glycoproteins and the glycolipids. So let's go on the first one, the phospholipids bilayer. It is the phospholipid they spontaneously forms into a bilayer, which is contained of the polar heads and the non-polar tails. So this is the polar heads, and these two are the non-polar tails, which is the fatty acid. Okay, so we have 2 types of fatty acid, which is the unsaturated fatty acid and the saturated fatty acid. If the type of fatty acid they use is the unsaturated, the membrane will be more fluid, and because they are these ones that were it wasn't straight, so that in the membrane they're gonna have more space for it, so it's kind of fluid. So this type of membranes they're gonna be better in the cold, because even in the cold they're gonna pack together. And the saturated fatty acid, they're gonna be better in the hot temperature because the membrane will be less fluid, because the membrane is less fluid. So yeah, see, as they're straight in a straight lines like that. Okay, so the next one is the membrane proteins. We have 4 of it in total, which is the peripheral proteins, the integral proteins, membrane protein, sorry, the transmembrane domain, and the pores. So the peripheral is the one that attached to the phospholipids from the outside. So yeah, from the outside, like this one, like that. And the integral is the one that is on the surface, like in the surface, like this one. See? It was in the surface, like literally in the surface. The third one is the transmembrane domain, the hydrophobic amino acid arranged in alpha helix, which is this one. Yeah, this one. Cause of this one they're gonna have the hydrophobic, which is doesn't like the water. Number 4 is pores. Pores is like these one. These are pores. They are the extensive non-polar region which within the transmembrane. The proteins can create a pores that allow water to pass by. So the membrane proteins, they have the various functions, and those various functions will come in a different appearance. So first one is the transporter. We can look at this photos. So the transporter is the one that will let the molecules from outside inside the cells. Next one is the enzyme, which is gonna I think it's gonna break those molecular to use make it easier to use. And the third one is the cell surface receptors. The fourth one is the cell surface identity markers. And then we have the cell-to-cell adhesion, which is the one that gonna join two cells together. And the last one is the attachment to the cytoskeletons. Yeah, so this one is the one that gonna attach to the cytoskeletons of the cells. So after we know the type or what we have in the membranes, next we're gonna know how the membrane transport things inside and outside. So first one we're gonna know about the passive transport, which is the one that doesn't require ATP. The first one is the diffusions, is the movement of the molecules from high concentrations to low concentrations. So this one they will continue doing the process until the concentrations is equal from the both sides. And since the phospholipid is hydrophobic, so the molecules that is using this process, the diffusions, must be small, must be in the small moleculars, or the other one is they can be in the non-polar, because as the phospholipids, they're non-polar, so the molecules that can get through the phospholipids should be the non-polars also. The second one is the facilitated diffusions. So this method is used for the molecules that can do the simple diffusion because of because they're non they sorry because of they're too big, or maybe another reason is because they are polar. So they need to use the proteins to help them in this process. So in this process, it's also moving from the high concentrations to low concentrations. So actually the diffusions, like the simple diffusions and the facilitated diffusions, they are quite similar. The difference is that the facilitated, they're gonna use the proteins to help the moleculars to get through the membrane. Okay, so the proteins, we have two type of proteins, which is the first one is the channel proteins. This one will allow the passage of ions, like ions, and the gate channels, they can open or close in response to stimulus, which can be both chemicals or electricals. So the the channel proteins, they're gonna be kind of like a tube where they're gonna let the molecular go through it, something like that. So there are three conditions to determines directions. First one is the relative concentrations on either side of membrane. Second is the voltage difference across membrane, and the third one is the gate channel, because gate channels can open or close like that. The second type of protein is the carrier proteins. This one will transport ions and some sugars and those amino acid. This one requires an concentrations difference across the membrane to use this one, and it must bind to the molecules they transport. So the thing is, it was not like a tube, like the channel proteins. They're kind of closed like that. The thing is, when the molecules coming through, they're gonna bind the molecular to themselves and then pull the molecular down into the cytoplasms. Then our third process, or the third method, is the osmosis, which is different. The difference from the last two methods is that the last two methods, they are moving the molecules to make the solution, no, sorry, to make the concentrations equals, right? But for this one, osmosis having the specifically molecules that they're gonna move, which is the solvent of the solutions, which the solvent of solutions is the water. So the water is gonna move from pure to less pure. For example, if we having a jellyfish and we moving the jellyfish, sorry, and if we put a jellyfish into the pure water, for example, like if we soak it into the water, the the what so the jellyfish they're gonna get swelled because the water from the outside they're getting into the jellyfish. As the jellyfish it's in the seawater, right? That's mean that it was less pure than the water that we use. So the water are getting inside the jellyfish. And there's in the osmotic concentrations, we have two different solutions. The first one is the hypotonic solutions, which is when there has a higher solute concentrations, and the hypotonic solutions, which has the lower solute concentrations. And if these two solutions they're having the same osmotic concentrations, the solutions will call as the isotonic. So we can look at this one. See, the hypertonic is the one that they will get like get shift, I think, because of the water are leaving the cells. See, so we can assume that the cells having is more pure than the things they're in because that the water leaving the cells. And for this one, we can assume also that the water outside or the solutions outside of the cell of the red blood cells, the solution outside must be pure. That's why the water get into the cells. And this one is the normal one, which is the isotonic. So, and then we have the osmotic pressure. It is force that used to stop the osmotic flow. So, the cell in the hypotonic solutions gain water, causing cells to swell, and this creating the pressures. If the membrane are strong enough, the cells will reach the counterbalance of the osmosis pressures, and the osmosis pressure will driving the water in, and the hydrostatic pressure will driving the water out without bursting. For example, like those plant cells, they can reach the counterbalance. But if the cells, if the membrane are not strong enough, they might burst, which is like those animal cells. So in this case, they won't have the counterbalance. Like we don't know like what is the maximums because they can't reach it. They are getting burst before they reach it. The next one is the active transport. So the active transport does require ATP, whether directly or indirectly. So they are against the concentrations gradient, which is they're going from low to high. That is why they're needing the ATP. So they are required to use the highly selective carrier proteins. So we have three types. The unipolar, moving one molecules at a time. Symporter, moving two molecules in the same directions, and antiporter, moving two molecules in opposite directions or different directions. Then the sodium-potassium pumps is the direct use of ATP for active transports, and the antiporter move the three sodiums plus out of the cells and two potassiums into the cells. So the positive transport made both sides equal, causing the NK pump to happen because when when both sides are equal, we need to do it again. That's why. And then we're having the coupled transport, which this one will use ATP indirectly, and they're using the supporters. So the molecules move against their concentrations gradients using energy stored in the gradients of the different molecules, like those glucose sodium supporters. They capture the energy from the sodium's diffusions to move the glucose against the concentrations gradients. And next one we're having the bulk transports. So this one does require ATP, but they're not active transports. So the first one is the endocytosis. So endocytosis is to transport the substance into the cells. We're having three types in total. The first one is the phagocytosis, is when the cells are taking the particular matter inside, mostly like solid things. The second one is the panocytosis. This one is when the cells want to move the fluid inside the cells. And the last one is the receptors immediate endocytosis. This one only takes specific molecules that bind to the receptors only. So if the target molecules they're binding to the receptors, so then they are going to accept that molecules. And the next one is the exocytosis. So this one is the opposite with the endocytosis. This one is to move the substance out of the cells. So like after the lysosomes digest those molecules, they're gonna move those products out. Next, the chapter 6. So we're gonna learn about energy as in metabolism. So energy, it is the capacities that to do works, which can come in many forms like heat, sound, and lights like that. So they have two stages. First one is the kinetics, which is the energy of motions, and the second one is the potentials, which is when it is the stage to store the energy. So heat is the most convenient way to measure energy. As a special note, I think. So the source of energy on Earth is the sun. Then the plant captures the energy to do photosynthesis. And next one we're gonna learn is about the metabolisms. So there's any chemical reactions that carry out by an organisms. We have two types again, which is the anabolic reactions, or the other name is the anabolisms. The catabolic reactions or catabolisms. So anabolic is to use the energies to build up the molecules, but the catabolism it is to release the energy as they break down the molecules. Oh, so catalysis, catalyst, it is a substance that make the chemical bonds with lower reactions, with the lower activations energy, making the reactions faster, where enzymes is the biological catalyst. See, it is the only biological catalyst. So, in metabolism, we have the redox reactions. So, redox reaction is an reductions reactions, which is how the energy transfers in molecular levels. So, the oxidation is when the atoms or the molecule lose the electrons, but in the opposite, the reduction is when the atoms or molecules gain the electrons. So these two are always be in pair. So the reactions, they're always be in pair. Next, we're having the law of thermodynamics. The first law of the thermodynamics is that the energy cannot be created or destroyed. Energies can only change from one to form another's. And the total amount of energy in the universe remain constant. During each conversation, some energy is lost as heat. The second law is that entropy or the disorder is continuously increased, not decreased, and the energy's transformations proceed spontaneously to convert matters from the more orders or like the less stable form to the less orders or the more stable form. So as we see, this is the examples. So to like it was the nature to happen like the disorderedness, but like to organize the disorderedness again, we need the energies to do it. Next is the free energy. So G is the energy available to do the work. So this is the chemical reactions. Yes, so the H stands for the enthalpy, which is the energies in the molecule's chemical bonds. And the T, it is the absolute temperatures, and S is the entropies, which is the unavailable energies. So after we are doing this process, we're gonna know if the delta G is the positive, the product's gonna have more free energy. It is gonna be the anabolic, and it's also gonna be the endergonic, and this requires an input of energies. But if it was the negative delta G's, they're gonna produce the less free energies. They're gonna be catabolic and also exergonic, and they're gonna release the free energies. So activation's energy is the extra energies that are required to starting the reactions. So exergonic reactions rate is the how much activation's energy is required. The rate can be increased by two ways. One is heating, and the second one is lowering the activation energy, which is the enzyme. So after we learn that, we're gonna know about the ATP, which is the cell's energies. So ATP stands for adenosine triphosphate. So it's composed of ribose, which is the 5 carbon sugars, the adenines, and of course the chain of 3 phosphates. That's the name, like the name. Sorry. So it is the key to energy storage, and the bonds are unstable also. So ADP is the form which has only 2 phosphate, and the AMP, which is the form which has only 1 phosphate, which is the lowest energy form. So the ATP cycles, ATP's hydrolysis gives out energies for energonic reactions. The couples reactions is the energies reactions by ATP is used by cells for energies required functions. Result is in the net G or the delta G's. So here's the pictures of the process of the ATP cycles. For example of those ATP cycle is the cell's respirations. So we're gonna learn next in the chapter 7, I think, about the cell's respirations. So next we're gonna learn about the enzymes, which most of the enzymes are proteins, but some of them can be RNA also. And the enzyme is the biological catalyst, which is work by lowering the activation energies. This stabilizes relationships between two substrates. So we're having the next one. We're having the active site, which is the pocket or the cleft, which for precisely for substrates to bind in, forming an enzyme substrate complex. And the enzyme's function is the rate of functions catalyze reactions that depends on the concentrations of substrates and enzyme. It can be changed by any chemicals or physical conditions that affect enzyme three dimensionals. So the things that can affect the enzyme functions is the coenzymes and the cofactors. Coenzyme is the one that work inside the enzymes. It is the one that needs to attach to the enzyme. But the cofactor is it needs to bond with the substrate to make it plug to the enzyme better. So it was helping. Next is the inhibitors. It is the substance that bind to enzyme and decrease its activities. So we have two types, which is the competitive inhibitors and the non-competitive inhibitors. The competitive is the one that compete with the substrates for active sites. So they're having the similar shape with the sites, like this one. See that like they're having the similar shape with the inhibitors so they can get in. And the non-competitive inhibitors, this one will bind to the enzyme at the sites at the other sites, which is the allosteric sites. So like it's normal to bind to that sites other than the active sites because like this one, the non-competitive, the shape of the non-competitive inhibitors, they're not similar to the active sites, so that's why they're going to bind to the allosteric sites instead. So we're gonna know more about the allosteric enzymes. So first is the allosteric inhibitors, which is this one. Actually, it's the non-competitive inhibitors, I think. So it's bind to the allosteric sites and reduce the enzyme's activities. So what is that? We're gonna know next here. So next one before that is the asteristic activator, which is the opposite with the inhibitors. So the activator is the one that binds to the asteristic sites, but it binds to increase the enzyme activities, not reduce. Next, we're gonna know about the feedback inhibitors. So feedback inhibitor is the end products bind to the first enzyme to stop producing the initial substrate, which is the negative feedback. So here, see as the initial substrate they get in, and then they produce this, and then they get in, and then they produce this, and then when they produce the last products, they're gonna fill up on the allosteric sites. So after they bind into the allosteric sites, they're gonna reduce the enzyme activities if they are the inhibitors. But if they are the activators, they're gonna make the enzymes continuously doing the substrate again and again, 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 like that. Okay, and next we're going to move on to the chapter 7. So first we're gonna know how the organisms obtains energies. So we have two ways. The first one is the autotroph, which is when the organism is producing their own organic molecules through the photosynthesis, like those plants. The next one is the heterotroph, which is when the organism is consuming the organic compounds or the organic molecules that made by others. So they're eating other organisms to obtain the organic compounds. And then we're gonna know about the solar respirations. So the solar respiration is the series of reactions where the energy is released in a small step, not all at once. Oh, and the little note is that the dehydrogenation. It is the lost electrons. It's about that the lost electrons are accompanied by the protons, which is the hydrogens. So this is the chemical reactions of the solar respirations. So the electrons, they're gonna carry the energies from one molecules to others. So the electrons carries, we have two. One is the NAD plus and the FAD, I think FAD. Yes. So NAD plus, it is the reduced form, which is the form that is ready to accept electrons. But in the reduced form, it is the in not H. So the NAD H is the one that is already accepts the electrons, same as the FAD, the fat. So fat is the form is the oxidized form, which is ready to accept electrons also, and the fat H2 is the form of the reduced form, which is the form that already accepts the electrons. So in the solar respirations, we have two of them, which is the respirations and the fermentations. So actually, we have three methods. I mean, three processes that they can do in the solar respirations. The first one is the aerobic respiration, which is the one that having the final electrons acceptors as the oxygens. So this one does requires oxygens, while these two doesn't. So at the anaerobic respiration, the final oxygen acceptor is the inorganic molecules, which is not the O2 or the oxygens. And the fermentation is when the last electrons acceptors is the organic molecules, which is converted into the lactic acid or the ethanols mixed with bind with the ethanol and the carbon dioxide. So first we're gonna mention about this first. See? And then after that we're gonna talk about this. So we're gonna talk about this later. We're gonna talk about the Arabic first. So before that we're gonna know about a bit more about ATP. So this is two mechanisms for synthesis of ATP. The first one is the substrate levels phosphorylation, which is to transfer phosphate group directly to ADP by using the enzymes, which is the kinesis, and during the glycolysis and Krebs cycles. So it's happened during the glycolysis and Krebs cycles. And the second mechanism is the oxidative phosphorylation. It is that like the ATP synthase use the energy from the proton's gradients. It's happened during the ETCs and the chemiosmosis. Chemiomosis, sorry. Sorry, chemiomosis. Yes, that's right. Chemiosmosis. So, a little notes for more informations. NADH is the coenzyme that has abilities to transfer electrons, like what I have said. And here, say it again. So, the aerobic respiration, solar respirations, it is the oxidations of glucose. So, we're doing this to oxidize the glucose into the ATP for our cells to use. So, we're having four steps. First one is the glycolysis, then the pyruvate oxidations, then Krebs cycles, and the last one is the electrons transport chain and chemiosmosis. So the aerobic solar respirations occurs in orders. So and the Krebs cycles and glycolysis create just a little ATP, but they are creating the crates on the electron carriers, which is those this one, the NaH, which they will meet up in the step 4, and they're gonna produce a lot of ATP. So the first step is the glycolysis. It is to convert the glucose to the more usable form called pyruvate. So this gonna occurs in the cytoplasms, and they doesn't need the oxygens. So they are converting one glucose, which is 6 carbons, into pyruvate, which is 3 carbons. So at first, the glucose they convert into from 6 carbons, they convert into 3 carbons and 3 carbons, but it was not stable. So they convert themselves again into the pyruvate acid or the pyruvate. So we are gonna having 2 pyruvates, and we're gonna having and the NAD plus turn into the NADH for the both sides. And also the ADP, they're gonna form out as the ATP. So we're having the 4 ATP in totals, but the thing is, from the start, we're using 2 ATP to break down the glucose also. So after we subtracting it, we're having 2 ATP in totals. Okay, so these are the end products that we have. If you guys are wondering why the NADH must be cycled, it must be recycled to keep the glycolysis running, because as the NADH or the NAD plus, they must return to the the NADH or the NADH must return into the NAD plus. And to recycle, we have 2 ways, which is the aerobic respirations and the fermentations. So we're gonna learn about that later, the fermentations. So the impact of oxygens. If there are oxygens present, the pyruvate is oxidized to the acetyl coenzyme A, which is the next step. And we're gonna doing the aerobic respiration. But if there are no oxygens present, the pyruvate is reduced in order to regenerate NAD plus into NADH, which is the process of the fermentation. So the second step is the pyruvate oxidations. So this one occurs, they move from the cytoplasm into the mitochondria. If this happens in the eukaryotes, but if in the prokaryotes, there occurs at the plasma membrane. So at first we are starting from the 2 pyruvate, which we receive from the last step, which is the glycolysis. Here we receive 2 pyruvic acid, the pyruvate. So we're using those 2 pyruvate to doing the pyruvate oxidations. So this step is when the pyruvate they're transferred into the mitochondria, right? And then the pyruvate they're converting themselves into the 2 acetyl coenzyme A. And in that process, they're having 2 carbon dioxides that getting released, and there are also 2 NADH, which are produced also in that meantime. After that, we're going to the Krebs cycles, which is the step 3. So after we getting the acetyl coenzyme A, we're gonna use it in this one. So the Krebs cycles there occurs in the mitochondrial matrix. The starting materials are only the 2 acetyl coenzyme A. So first of all, we're gonna have the 4 carbons molecules, which is the oxaloacetate. So it's the 4 carbons molecules. So we're having it at first, and then the acetyl coenzyme A from the pyruvate oxidations, they bind into the 4 carbons molecules, this one. oxaloacetates. So after they binding together, the oxaloacetates turn into the citrates. But in the meantime, as the acetyl coenzyme A, like they bind into the oxaloacetates, the coenzyme A is bounced off, and they are binding together into citrates, which is the 6 carbon molecules. So after we having the citrates, they're going to do the process again and again, um, which is to release, yes, which is to release the carbon dioxide out. But while they're releasing the carbon dioxide out, they're also making the NAD plus into NADH from nodules. So after they doing it in the first time, after they release the carbon dioxide, the citrates they're turning into the 5 carbon molecules, and then they're doing it again, so it's turning to the 4 carbon molecules. And while they're doing the process, the ADP plus phosphate they're turning to ATP also. So lastly, they're producing 1 ATP. And as their 2 Krebs cycles happens, so the amount of the molecules that I have been saying is doubled. So the NADH is doubled and also the ATP. So at this point, after we are going through 3 steps, so we're gonna look in the total molecules that we have, which is like the total things that glucose has been oxidized into. So first is we're having 6 carbon dioxides, we're having 4 ATP, we're having 10 NADH, and we're having 2 fat H2. So these electrons carriers will proceed to the electrons transport chain, then produce a lot of ATP later. So here it is, the last steps, the electrons transport chain and chemiosmosis. So this one is happening inside the inner mitochondrial membrane, which is the crestae in the eukaryotes. In the prokaryotes, they're happening in the plasma membrane. So the starting material is the NADH, the fat H2, and the oxygens. So this one is the step that we require the oxygens from all of it, but it's very important because if we don't have oxygen, we can't do all of this. So, and the end product is gonna be 32 ATP, water H2O, of course, and NAD plus and fat plus. So, the process is that the electrons from the NADH and fat H2 transfers to complex of the ATC, which is the series of membranes-bound electrons carriers. So, then they're releasing the H plus out to the inner mitochondrial membrane. See? So they're here and then they are releasing the H plus they're releasing the H plus out from the channels. And in the meantime they're also releasing 2 electrons. See? They're releasing 2 electrons. Also the fat H2, they're doing that also. But they're only releasing 2 electrons. They didn't release the H plus. So those electrons are gonna gather together. They're gonna gather together to the next steps. Oh, so before going to the next step, the oh, sorry. So I was look wrong. I was yeah. So those electrons gather together to here, and then they're gonna bind with this one, 2H plus and 1/2 of the oxygens. After they are binding together with those electrons from the net H and the fetch H2, they're gonna process out as the waters. This is it. Yeah. And lastly, after after we clear the electrons out already, the H plus, oh, sorry, the H plus they're gonna gather together. So this one, like after they have been pumped out of the cells, like of the mitochondria, and it's gathered together at other sides. So then it's mean that those H plus they're gonna gather in this side for too much, right? And after they gather in that side for too much, they're gonna cause the diffusions because the concentrations are not the same in the two sides, right? So they're causing the diffusions. So those H plus gonna get back into the matrix by the ATP synthase. Uh, Here, this one, the ATP synthase. And after those H plus get back into the ATP synthase, that's those H plus gonna combine with the ADP plus phosphate, and that's gonna produce the ATP. So this is the process. So this is all the process together. So glucose, and then the private oxidations, and then Krebs cycles, and then the last one here, the things that we have learned, the electrons transport chain and the chemiosmosis. So, but like there's like in different books they are saying the different amount of TPP per glucose. So some says that there are about like 36 to 38 ATP. Some say that it was 30 ATP, and some also say that it was 32. Like 30 for prokaryote, sorry, and 32 for sorry, I'm so sorry. So 30 ATP for the eukaryotes and 32 ATP for the prokaryotes. Okay. Then we're gonna learn about the oxidations without oxygens. So, yeah, so like without oxygens, it can happen two things, which is the anaerobic respiration and the fermentations. Anaerobic respirations is the final electrons accepted is an inorganic molecules, not so it's not the oxygens, and many prokaryotes use sulfurs, nitrates, carbon dioxide, or even those inorganic materials. But in the fermentations, the final electrons acceptors is an organic molecules, and they're gonna convert into the lactic acid and the ethanols. The anaerobic respirations is the anaerobic respiration. It is the we we can have two things, which is the methanogens. So in this one, the carbon dioxide is used instead of the oxygens, and the carbon dioxide is reduced into the methane. This one is found in diverse organisms, includes like those cows. And this one, the sulfur bacteria. The inorganic sulfate is reduced. In this one, the inorganic sulfate is reducing into the hydrogen sulfate. And the early sulfate reducer sets the stage for the evolutions of photosynthesis. Next topic is the fermentations. See, after we have finished this one, so the last one of the cellular respiration is the fermentation, which is what we're gonna learn. So, in fermentations, after the glycolysis occurs, there's no oxygens, which is the final electron acceptors, right? Meaning that they can't do the next step, like those pyruvate and those Krebs cycles, those things, because as they doesn't have the oxygen, so there won't be any final there won't be any final electron acceptors. so they can't do it same as the aerobic respirations. So they're gonna doing instead of that they're gonna doing it into two types. There are two types that two ways that they can do the fermentations. The first one is the lactic acid fermentations, which the process is that the pyruvate they're gonna change from into the lactic acid. In this process, they're gonna use electrons from NAD plus, NAD H, meaning that the NAD H will drop E at the E or the electrons, the E or the electrons off, and they're gonna become the NAD plus again. See, as the pyruvate become lactate, they're gonna the NAD H they're gonna drop off the E, which is the electrons. So this is the start product, and this is the end product. Then the next one is the ethanol fermentation. The process is that the pyruvate change form into the acid, uh, acetal, acetal, acetaldehyde, and they're gonna release carbon dioxide. Then NADH give out the electrons at acetaldehyde and to form the ethanols. And the NADH gonna return back into the NAD+ again. So this gonna be the start product, and this gonna be the end product. The last chapter is the chapter 8. So in this chapter, we're gonna learn about the cell's divisions. So to continue the life of living things, it is based on reproductions of cells or the cell's divisions. In unicellular organisms, divisions of one cell reproduce the entire organisms, but in the multicellular organisms, depends on cell divisions for development from the fertilized cells to growth and repair. So first type is the binary fissions. So this happens in those prokaryotes. So it's a type of cell division that the chromosomes or like the cell walls and the cell membranes from the origin cells move apart very fast, like those cancers, like bacteria and tumors. Since the prokaryotes evolved before the eukaryotes, so mitosis probably involved from the binary fissions. And the other name of the binary fissions they're called as the amitosis. Next is after we learning about the prokaryotes cell divisions, right? The next one is the eukaryote cell divisions. So we're having the mitosis, which is the divisions in nucleus. And then the cytokinesis is the divisions in the cytoplasms, which the mitosis happens with the non-reproductive cells or the somatic cells. But the meiosis is how the gametes produce, which is needs for like those sexual reproductions, which is using for the reproductive cells. And then the cell cycles. In 1882, the German anatomist named Walter Fleming developed dyes to observe chromosomes during the mitosis and the cytokinesis. So the cycle consists of first one is the mitotic with the M phase. It is the mitosis and the cytokinesis phase. Next one is the interphase. So interphase is the preparations for cell divisions, which takes about like 90% of the cycles. it can be divided to the cell phase, which is the G1, the first gap, the S phase, which is the synthesis. This when the chromosomes duplicate itself, and the last phase is the G2. So as you see, but if they're gonna be a checkpoint around here, so if they doesn't pass the checkpoints, which is in another meaning is that they're in the G0 state. They're gonna left the cycle because they can't do the divisions, like the cell divisions anymore. So the mitosis phase, these are the phase, like the stage of the mitosis phase. First one is the prophase. So the prophase is when the chromatins develop into the chromosome, which is two chromatids that's held together at the kinetochore became vestibules. As you see, like at first, like the chromatins, they are like unclear, right? But then in the prophase, they started to be clearly and like more vestibules as the chromosomes. So next is the prometaphase. This is when the nucleus started to disappear. As you see here, they started to disappear, right? Then the next state is the metaphase. The chromosome will start to line up into a straight line with the help of the mitotic spindle, which we're gonna learn next. And the anaphase is to separate the chromosome into the chromatids by pulling its two water poles. As you see, after they line up at the metaphase plate, at the anaphase stage, they're gonna separate those chromosomes, which we're gonna call that as the daughter chromosomes. And then having the telophase, which is the phase that the nucleus envelope reforming again around each chromosomes. And the last one is the cytokinesis, which is when the cells start to separate, like it started to separate the cells completely. Mitotic spindles. It is the material that helps to control the chromosome movement during the mitosis. So the process starts from the G2. The two centrosomes, they're gonna stay at rest here, as you see. Then in the prophase, the two centrosomes, they're starting to extend shortly, called as the asters. As you see, they're starting to extend, and they're also starting to extend the spindles already. Some spindles. Oh, then in the prometaphase is when the centrosomes move to the opposite poles and develop the aster into the spindle fibers. So, like first, the centrosomes are here. This one gonna move to the poles. So they're gonna be opposite to each other's, like that. In opposite pole of each other's. Sorry. And then the metaphase is when the spindle's fiber attached to the kinetochores, which is the proteins in the middles of the chromosomes. So they're gonna connect to the kinetochores and move the chromosomes into the line. And the last one is the anaphase, when the sister chromatids separate and move along it's gonna move along the kinetochores microtubules. So that's all for the mitosis. This is all for it. Next, we're gonna go with the meiosis. So the meiosis, first we're gonna have two process. The first one is the prophase one. The prophase one occupies around 90% of the time required for meiosis. The chromosomes begin to get condensed, and each pair of the chromosomes form a tetrad. The tetrad is the group of four chromatids forming together. And in each tetrad, the chromatas, the chiasmata occurs, which is the crossing point the crossing over points. As you see here, this is the chiasmata. As you see, they're getting crossed over, right? Like that. So, and in the crossing over, the non-sister chromatids exchange DNA segments. As you see here, they exchanging, see, these blue chromosomes, they exchange their legs. So they are having the legs from the pink one, and the pink one having the legs from the blue one also. The next stage is the metaphase 1. Metaphase 1 is that the tetrates they line up at the metaphase plate and with one chromosomes facing each poles, and the microtubules from each poles are attaching to the kinetochores of one chromosomes of the each tetrads, and the microtubules from each poles are attached to the kinetochores. Then the anaphase 1. In anaphase 1, the pairs of homologous chromosomes separates. So each chromosomes move towards each pole, and the sister chromatids remain attached. The telo-1 and the kinetosis. In the beginning of the telophase 1, each half of the cells has a diploid set of chromosomes, meaning that each chromosome there still consists of the two sisters chromatids, as like they're having four. They're having two, right? That's mean that when they get separated, they're still having four. See, four. So like it was like this. Sorry, wait. It's like this, right? Then this is one. This is one. This is one, and this is one. Four. So totally four. Four, or like two sister chromatids. The cytokinesis framing two haploid daughter cells. So in the animal cells, the cleavage ferrule forms, but in the plant cells, the cells plate forms. So in this one, no chromosomes replications occurs at the end of the meiosis one because the replication already happens. Yeah, at first it already happens already in the S phase of the interface. Then we're having the meiosis two. So the meiosis two, they're gonna happen twice as at the end of here. You can see the pictures because at the end of the meiosis one, the cells divide into two. So this is the cell at first. They divide into two. As this one is the meiosis one, right? So they have been divided into two cells. So meaning that after the meiosis two, they're gonna divide into four. Because here they're gonna divide right into two, and this one gonna divide into two. So we have in total four. Yes, but the thing is, the meiosis two, the process is similar, like very similar. Actually, like the same process with the mitosis, but we can't call it mitosis. It is the meiosis two. So I won't be writing the process as it is the same process with this one. Yeah. So this is the pictures. So this is the meiosis one, as you see here. They're crossing over, having chiasmatas line up. Here they are separating, but when they're separating, they're still in a chromosomes form because they're having four chromatids at first, and then here they're starting to separate, and then they're doing. It is the meiosis two, but they're doing the same process with the mitosis, like exactly the same. Next, the last topic is the genetics variations. So, genetics variations is the sexual productions result in the infinite possibilities of genetic variations. Because of this, each offsprings have their own, like having the unique genetics. The three methods which genetic variation is promote. One is the independent assortment of chromosomes. So when the chromosomes line up on the metaphase plate randomly at the metaphase 1 of meiosis, the number of combinations possible when the chromosomes are sorted independently into gametes is 2n, which n is the haploid numbers. So, which for the humans, there are more than 8 million possible combinations of chromosomes can happen. So 8 millions are formed to กำลัง23 yeah. So n it's the here it is 2n, right? So n for human it is 23. The next method is the crossing over. Crossing over occurs in the prophase 1 of the meiosis and is when the homologous chromosomes having paired together, and they swap the segment of the DNA via chiasmatas, and therefore exchanging the genetic materials, increasing the diversity of the gametes. And the last way is the random fertilizations. So random fertilizations also promotes genetic diversity, as any one of the millions of the sperm haploids produced and can, like those sperms, all of them can fertilize the specific 8 eggs that was released out of the millions of the female has. So like the million sperm from the boys, from like the man and from the male, they're gonna be random selects to match with the one of the millions eggs from females also. So it was so random, right? So there can be many case possibles happen. So these are all informations and topic about from chapter 5 to chapter 8 in biologicals in M4 for the finals in semester ones. So thank you for watching and giving your attentions.